sama berapa bulan? Dia. Aku tidak cerita lagi tu, tapi macam berapa bulan ke?
Tak tahulah, macam lain je daripada tadi.
Sebabnya dia cuti.
Tolong leave je lah. Dia macam biasa pun.
Hello, good afternoon. We're very sorry for the technical glitch just now. Okay, the link was expired, so we had to create new link right away. So apology for that. Just a few housekeeping. Could everyone please mute your mic? Okay, and then during presentation, also could everyone turn off your camera. Okay, during the Q&A, question and answer, the presenters should turn on the camera. Okay, provided if your internet is smooth. So please turn on your camera and answer your question. For the rest, please just keep the videos off. All right. Okay. Thank you for your cooperation. We will start in a while. Okay, in a bit.
Okay.
Okay, Assalamualaikum and very good afternoon to everyone. We hope that all the presenters are here. So for this afternoon, I will be chairing this session. Okay, my name is Dr. Satirah Zainalabidin from UKM. I will be one of the judges for this afternoon. And with me this afternoon, we have Dr. Vinod. Okay, he is an expert in molecular virology from Monash University in Malaysia. Very pleased to have you here with us, Dr. Vinod. Hi there. So without further ado, we will start with the presenter. Yeah? Sorry. So PCA 04, Salmah Sedek, you are here, right? Okay, Salmah Sedek. Okay, we'll start with her presentation.
Hello everyone, I'm Salmah Sedek from International Islamic University Malaysia, Kuantan. So today I'm glad to be able to present my research on the effects of different coating substrates on human primary lung cancer cells group. So briefly, similar with other adherent cells, primary lung cancer cells are encouraged dependent and they need to attach to the extracellular matrix for them to be able to grow, proliferate, and survive. So therefore, it is very important to find the right coating substrate that will help with growing and survival of these primary lung cancer cells. So my research involved collecting lung cancer biopsy specimens from Hospital Tengku Afwan Hassan, Kuantan. So far we have been able to collect around 40 biopsies, lung cancer biopsy specimens. After the biopsy collection, cells were immediately brought to the lab for cell isolation. So after cells were isolated mechanically, these cells were incubated with special type of medium, the airway epithelial medium, supplemented with 10% of fetal bovine serum and 1% of penicillin and streptomycin. So these cells were then incubated in 3 different types of coating substrates, which were collagen, type 1, polylysine, and gelatin. And then after 14 days of incubation, the morphology and the growth state of these cells were observed under inverted microscope. So as you can see from figure 1, only the cells with collagen type 1 are coated plus have shown the epithelial-like morphology and monolayer attachment, while other cells with gelatin, polylysine, and non-coated plus do not show any growth. This interesting finding has helped us a lot in growing our human primary lung cancer cells. And as of now, it is good to know that collagen type 1 coating solution managed to help us with the, growing our human primary lung cancer cells. Thank you so much.
All right, thank you very much, Ms. Salmah Sedek. Are you here?
Yes, doctor. I'm sorry, I cannot turn on the video.
Right.
My internet is not good.
Okay, all right. Just one question for me. How has this method been established before?
Some of the establishment of cancer cells, the primary cancer cells particularly, have used collagen type 1 before, but they haven't tried with different kinds of coating substrates. In terms of our research, we tried with collagen, polylysine, gelatin, and also the non-coated substrate, non-coated plus as the control. So I can say the other studies have some of the coating substrates, but not all of the coating substrate that we have been using.
All right, so your novelty would be on the coating substance. I was going into the novelty of the study. Sorry again, housekeeping. Could everyone please turn off your microphone and just let the presenter answer the question? Okay, please, Ms. Salmah.
Yes. Again, the second question, doctor.
I was asking about the novelty of your study.
Oh yeah. This part of the coating substrates that we have been using is only like one part of the study that we have been doing. So other than that, we also made the research focused on the type of culture medium that we used, and also the
All right. Okay, it's okay. Thank you very much, Ms. Salmah, for answering the question. Okay, so we shall proceed to the next presenter, Ms. Maziana Binti Mahamood from UITM. Okay, please welcome.
Assalamualaikum and good day everyone. My name is Maziana Mahamood from Faculty of Pharmacy UITM. Today I would like to present my research finding with the title The Effects of Vitamin E and Vitamin C Supplementation on Antioxidant Enzymes in Down Syndrome. The copper zinc superoxide dismutase SOD gene is present on chromosome 21. Therefore, individuals with Down syndrome may be under oxidative stress. Antioxidant supplementation are thought to decrease the oxidative stress level in these individuals. Therefore, the objective of this study was to determine the effects of tocotrienol-rich version, TRF, and vitamin C supplementation on antioxidant enzymes in Down syndrome individuals. 60 individuals aged between 2 to 29 years old with Down syndrome were recruited and randomly assigned to receive either supplementation with 150 mg TRF and vitamin C 250 mg or placebo. Blood samples were obtained from each subject at baseline after 3 and 6 months of supplementation for the measurement of antioxidant enzyme, superoxide dismutase, catalase, and glutathione peroxidase. Changes in the activities of antioxidant enzyme with TRF and vitamin C were not observed when comparing the baseline to 3 and 6 months for SOD activity as well as in catalase and GPX. There were also no association between the SOD catalase and GPX activities and age in both treatment and control groups. In summary, it can be concluded that there was no significant difference in the levels of antioxidant enzymes, and further study should be conducted in order to understand the mechanism that contributed to this condition. Thank you.
Okay. Thank you, Maziana, for your presentation. Are you here?
Yes, I'm here.
Fantastic. Just want to ask, what is the rationale of using vitamin E in combination with vitamin C? Were there any studies done before, to look at the synergy of both of these vitamins in reducing that particular enzyme? What made you do this experiment?
Yeah. Thank you, Dr. Vinod, for the question. All right. Basically, the reason why we used the combination of vitamin C and vitamin E in this study because of the mechanism, the mechanistic action between these two vitamins. For example, we know that for a long time that use of antioxidant, especially for vitamin E, during the process of lipid peroxidation, we know that from vitamin E, the peroxyl radicals are formed. So in the first step in the scavenging of peroxyl radicals, the lipophilic antioxidant of vitamin E will occur. Therefore, the chemical reaction between these reactive radical will be transformed into a non-radical, and then with the help of vitamin C, this radical will be taken over by vitamin C in the plasma. Therefore, the vitamin C radical might react with NADH to regenerate vitamin C, and this reaction catalyzed by the ascorbate free radical reductase. Therefore, at the end, the reactivity of this radical is totally absorbed in these three basic steps. So, previous study have shown that this what we call it this action between this combination of vitamin C and vitamin E. That's the reason, the main reason why we choose vitamin C and vitamin E in this study.
Okay, thanks. You have explained nicely how this mechanism of antioxidant happens, but you did not see it. Why is that in your experiment, in your opinion?
All right. So basically, in general, the use of vitamin E and vitamin C is quite known supplement that we in the market, and also people always use this combination. So another reason why we choose this is because of the participant or the subjects in this study is Down syndrome. So it might affect the participation of the subject in this study if we use the unfamiliar supplements or other types of supplement, because that's the reason lah. That's the reason why, another reason why we choose vitamin E. It's a common supplement in the market.
Okay, thanks.
Okay, thank you, Ms. Maziana. So we'll proceed to the next presenter, Ms. Fazilah. Okay, from Department of Physiology, you can.
Welcome to my presentation. My name is Fazilah Binti Abdul Hamid, Master Student from HUKM. The topic of my presentation is investigating the exploration of endometrial resistivity markers in women with polycystic ovary syndrome following progesterone therapy. Let's start with PCOS. PCOS, which can occur due to several factors including insulin resistance and obesity, and might be cause implantation failure. Now, let's move on to my next topic, which is several steps that have been taken throughout the study running for 1 year. As you can see on this methodology, a total of 40 women range in age 26 to 45 years, and all subjects were divided into 2 groups, which were classified with PCOS and control group. In the PCOS group, daily oral micronized progesterone was monitored for 10 days, and the control group was monitored for normal regular menses and ovulation. Blood and endometrial tissue biopsy collected in all groups. After that, the endometrial biopsy was collected during the mid-secretary phase, which occurs 7 to 9 days after ovulation. The main 3 points, we find that FSH, estradiol and progesterone levels were statistically lower in PCOS group than in the control group. Second, LHE and DHEA level of PCOS group were not statistically different from the control group. And the last one, not significant change we found in the endometrial integrin, heparin binding, MECY1, and E. carrying expression in Boots group. To conclude, I would like to say endometrial integrin, heparin binding, MECY1, and E. carrying expression doesn't not differ between PCOS and normal women, suggesting further study to discover the relation of the genes to PCOS women. Thank you very much for your times.
Okay, hi, Fazilah.
Hi.
You're here, right? Yeah, hi. Thank you for your presentation. Nice presentation. Why you think there are no differences in your gene expression? That's number one. Number two, did you look at protein levels of those corresponding transcripts?
The gene expression, we have the 4 gene expression, hyperimandine, integrin, MECY1, and E. carries, but not significantly PCOS compared to the control group. And
why? My question is, why would there no difference? In your opinion?
oh, my opinion.
Okay. Yeah.
My opinion. I was read one paper. My opinion is maybe not enough the dose of the utrogestan is progesterone to the PCOS patients. I think that's why my research is not significantly than the control groups.
Okay. Did you also look at the protein levels? Did you quantitate the protein levels of those markers?
Again, doctor?
Did you observe or did you quantitate the protein levels of those markers, the samples you obtained? Did you look at the protein levels?
Protein levels?
Yeah, of those markers.
Oh, protein levels. Actually, I do the protein level, but the same result not significant for the boost of groups.
Okay, thank you.
Okay, thank you.
Okay, thank you, Ms. Fazila. We move on to the next presenter, Ms. Nuraina Mezlan. Please welcome. And sorry, can I just please make sure that Nuraina Fatiha is here? I hope you're here.
Yes.
Okay, great. Thank you.
Hello, I am Nuraina Fatia Mezlan. I want to present on the research entitled Internet Gaming Disorder, Prevalence and its Effect on Insomnia and Psychological Distress among adult gamers in Malaysia. Nowadays, there are many adults spend too much time playing online games, especially in this pandemic era, and it's very
Yes, I'm
concerning.
here.
So, this study is conducted to investigate the prevalence of internet gaming disorder among adult
Yes.
gamers in Malaysia and its
Hello.
association with insomnia and psychological distress, which are depression, anxiety, and stress. So the
Yes,
method
I'm
used in
here.
this study is cross-sectional survey via Google Form, and we use purposive sampling as sampling method. The questionnaire is made up from 3 validated questionnaire, which are Internet Gaming Disorder Short Scale Form to determine the IGD, Atten Insomnia Scale to determine the insomnia, Depression, Anxiety, Stress Scale 21 to determine the severity of depression, anxiety, and stress. So from the results, from table 1, we can see that from the total 280 respondents, there are 10 respondents having IGD. So it made the prevalence of the IGD among adult gamers in Malaysia is 3.6%. And this value is not far from some other countries, which are Korea 4% and German 5.7%. From the table 2, Pearson correlation table, the IGD significantly associated with insomnia, depression, anxiety, and stress, with R value 0.144 and 0.202, and P value below 0.05 and 0.01. So the conclusion is the prevalence of IGD among adult gamers in Malaysia is low. However, it is also associated with insomnia, depression, anxiety, and stress. So it needs to be given attention by public and healthcare professionals. Thank you.
Okay, thank you, Ms. Nuraina. Current study about internet gaming disorder. What is your suggestion for further study? Like based from your conclusion, okay, what is your
Here you can off your video and then try.
Nuraina, if we lost. Hello, Nuraina.
Yeah, it's audible now.
Okay. If Nuraina is not around, then we'll just move on to the next speaker. The next presenter, Mohammad Hassan from Universiti. Are you around?
Yes, Doctor, I'm here.
Okay, great. Okay, so let's play your presentation.
Good morning, everyone. Myself, Mohammad Hassan, and my topic is my spade, a Malaysian tool for Streptococcus pneumoniae, arches, diagnostics, and evaluation. Everyone knows Streptococcus pneumoniae is the leading causative agent of meningitis, community-acquired pneumonia, otitis media. Burden is increasing in society in recent years, and its assessment on time is needed to overcome its burden in society. That's why we plan to um, develop a tool which can assess, which can do assessment like professionals and help professionals to for selection of antibiotics according to the patient's condition and severity of pneumonia. Here is a interface, and you can see it's very easy to use. Just click on button bad, and you can you have to select some your symptoms which are you are facing, and at the end you will get a report in which you can see it's a cases. You have to see in which case you lie, and according to which case you should be treated your patient, and which antibiotic will be effective in that case. It's the new novelty of this tool that we develop a tool which can not only check severity level of severity index of pneumonia, but also suggest which antibiotic is acceptable or will be effective, and which how much dose we have to give it. It can help professionals as well as learners.
Okay, thank you, Muhammad Hassan. I'm sorry, I was kind of lost. What was the conclusion of your study?
Yes, doctor.
Was it a review study?
It's actually doctor. It's actually a tool, Android tool, in which you can assess your patient's severity level of pneumonia. It's kind of tool in which you have to select its symptoms of your patients, and as well as their laboratory reports like arterial pH and everything. And at the end, you will get some results on basis of the scoring algorithm which we develop. And at the end, you will get results on the basis of these algorithm, that how your patient is in high mortality rate or in low mortality rate, and you can decide which antibiotic is suitable for your patient.
All right, okay. All right, thank you very much. Okay, nice work. Okay, so we move on to the next presenter by Miss Chen. Chen from Monash University. Okay, please welcome.
Good day, everyone. My name is Winning, and I'm presenting my project titled The Evaluation of Anticonvulsant Potential in Otosiphon Staminus, also known as Kucing, using a mice seizure experimental model. As we know, epilepsy patients have recurrent seizures, and the currently available anti-seizure drugs are proven to provide symptomatic relief and may have adverse effects on patients. Hence, we seek to discover novel therapeutics, such as using natural plants or extracts. Otosiphon staminus (OS) is known to have beneficial bioactivity and have been shown to reduce seizures in zebrafish. Thus, we decide to probe for possible anticonvulsive properties using mice seizure model. We conducted repeated dosing in mice using 50, 100, and 200 mg per kilogram of OS for 7 days. Subsequently, we induced seizures mechanically using maximal electroshock seizure model (MES) and chemically through pentylene tetrazole (PTZ) model to determine if OS can shorten the duration of hind limb extension and delay the first and second onset of seizures. Unfortunately, our results revealed no significant difference in any of the treatment groups when compared to the control of both models. However, we believe that further extension of this study could be done by including a chronic model through long-term administration or dosing, and detailed mechanistic studies of the potential anticonvulsive properties in OS. Thank you.
Hi, Lincoln. Thanks. You're here, right?
Yep, I'm here.
Nice study, elegant. Just one question. You suggested some improvements in your study, right? Can you elaborate a bit on how those improvements actually, will give you different results than what you got from your first, there were not much significant changes, but with these improvements, how you think it will change? Will it change?
Yeah, so actually when you're using like a natural extract or product, it's a combination of so many different things in that one product. So it would be better if you could actually isolate the compounds that are that could contribute to that specific bioactivity. So also for using natural products or extract, sometimes their effects may not be as immediate or as compared to synthetic medication. So that's why I propose to have a longer term of dosing for the mice. Yeah.
Okay, thank you. Just one last question. Were there any studies before to look at this particular plant compound, Misa Kuching? Because I've read a few studies, interesting studies that properties, pharmacological properties of this compound.
Yeah, so there are a few studies, but they were mainly focused on antioxidant, anti-inflammatory, and anti-diabetic studies. So yeah, there were a couple of them. And as for the lab which I conducted this, they actually used it in zebrafish as well to look for anti-seizure activity. So I would say it's pretty quite a well-known product. It's just that we would need further studies to, to uncover its properties. Yeah.
In the zebrafish model in the lab you mentioned, right? Were there any different results obtained when compared to the mice?
Yeah, so surprisingly in zebrafish, they actually demonstrated anti-seizure activity. So though I'm not entirely sure what's the difference, but apart from the species variation, but it's definitely interesting and should be looked into more to compare the differences. Yeah.
Definitely. Thank you so much. Thank you.
Yeah, sure.
Thank you.
Okay, thank you, Winning. So, Miss Nuraina, are you here before we go too far off from you? Are you around?
Yes.
Okay, all right. Thank you for your presentation. I was asking, so in your conclusion, you mentioned that actually the prevalence of internet gaming disorder in Malaysia is considered low, right?
Low.
Was that what was your conclusion again? Hello, are you here? Got disconnected again. All right, Nuraina. Okay, it's okay. So we'll just move on again to the next presenter by Yee Yong Mei from Utah. Okay, please welcome.
Hi, the topic of my study is hypotensive effect of an partially purified fraction of Ganura procumbens in spontaneously hypotensive rats. Ganura procumbens is a fast-growing medicinal plant that is anecdotally used for treating hypertension. Previous studies have shown that a partially purified fraction of Ganua procumbens, FA1, is able to inhibit the activity of angiotensin converting enzyme ACE in vitro, where inhibition of ACE is well known for decreasing the blood pressure, BP, of an individual. Therefore, in this study, we aim to investigate the potential hypotensive effect of FA1 in the SHR and animal model of hypertension. Age-matched WKY rats were taken as control for SHR. To begin with, the right jugular vein was cannulated for intravenous bolus administration of FA1 at different doses from 0 to 10 mg per kg, while left carotid artery was cannulated for continuous measurement of BP. Changes in the BP due to the administration of FA1 were obtained by calculating the differences between BP before and the lowest BP recorded after the administration. The result shows that intravenous bolus injection of FA1 caused a dose-dependent significant reduction in MAP, mean arterial pressure, in both anesthetized SHR and WKY rats. The reduction in the SHR was significantly greater than in the SHR rat, notably at the higher dose of 10 mg per kg. Hence, we conclude that FA1 does exert a hypotensive effect in the SHR rats, possibly in part due to the presence of ACE inhibitors, and our future direction is to perform phytochemical screening of FA1 to identify the bioactive compound with hypotensive properties. Thank you.
Okay, thank you, Miss Yong. Are you here?
Yes, I'm here.
Okay, so interesting study on the hypotensive effect, yeah? So you were measuring the blood pressure through the direct method, okay,
Yes.
of blood pressure. So did you, what is this FA? Is it the crude extract?
It's not a crude extract, it's a partially purified extract.
Okay, so you're suggesting that it's through the ACE pathway, yeah?
Yes.
You're suggesting it works through ACE inhibitor pathway, but
Yes.
you did not know for sure. So how can you confirm that in the future study?
Okay. So I'll do a few experiments in the future study. So one is I'll need to run another ACE1 inhibitory experiment in the future, and also actually the ACE inhibition activity of this partially purified fraction has already been studied and shown by previous studies. So we can partially conclude that this actually the Gardnerella procumbens partially purified extract does exert the hypotensive effect through the ACE inhibitory activity. One is by the previous study, and another one is that I'll be doing additional experiment, the in vitro experiment, as well as supported by possibly in silico study.
Okay, all right. Just one last question. So you did not, what is your opinion about doing another, let's say, another group with ACE inhibitor in your study?
Another control in ACE inhibitor?
Yeah, another positive control. Let's say you test with ACE inhibitor and you measure the blood pressure.
Or you mean incorporate a positive control in this in vivo study, is it?
Yes.
Yeah, that would be better actually, so that we can compare the effect of this extract with the positive control drug to see how much effective of this extract as compared to the positive control drug.
All right. Okay. Thank you very much.
Thank you very much.
Okay. Nice study. Okay. Next, we move on to Miss Huma Shahzad from IMU. So please let us hear your presentation.
Hello, everyone. The title of my poster presentation is Bergenin Protects Hyperglycemic Induced Testicular Oxidative Stress and Reformed Steroidogenesis in Streptozotocin-Induced Type 2 Diabetes Rats. This study basically was conducted in the diabetic rats in which there is a damage to the testicular tissues because of the oxidative stress. Uh, we hypothesize that because of the diabetes and oxidative stress, there is a damage to many tissues and organs, and it also affects the testicular functions in the males. The berganine helps to improve oxidative stress by lowering the hyperglycemia because it has shown in many studies that berganine proves to be a good berganine helps to improve the oxidative stress in the diabetes. So we hypothesized that berganine could also prevent the oxidative stress or hyperglycemic induced oxidative damage in the testicular tissues of the diabetic rats as well. So in the material and methods, we induce diabetes mellitus by giving the high-fat diet to the male rats for about 12 weeks. And at the end of the treatment, we give them the single streptozotocin injections. And after that, we also measure the fasting glucose levels. And once we confirm that the rats are diabetes, then we divide them into further groups. And of course, we give also the treatment to the diabetic group in a high dose and the low dose of the berganine. So in the result part, as you can see, the sperm count and viability increases, and also the inflammatory markers there were highly markedly visible in the diabetic groups as compared to the groups which receives the berganine treatment at low and high dose groups. And also the level of steroidogenesis is improving in the berganine at high doses, and also the apoptosis markers activity reduces in the treatment group which receives the berganine at high doses. So in conclusion, we conclude that diabetes the berganine can help to improve, can alleviate the effect of oxidative stress in the diabetes, in type 2 diabetes. Thank you.
Hi, Huma. Thank you. Are you here?
Hi, Dr. Vinod. Yes, I'm here.
Thank you. I think your study looks brilliant. Can you talk? Can you elaborate a bit more on this compound berganine?
Yes,
Anything
sure.
about it? Yeah.
Yeah,
Yeah.
actually, the berganine is a famous compound, and
Mhm.
it's more famous in India, I think, because
Mhm.
I read about it a lot. That it is a form of Ayurveda as well. It is known as
Mhm.
Ayurveda
Mhm.
because they extract it from many plants. Like it can be found in some of the common, species, botanical species. The names are very difficult to pronounce, but this berganine compound is actually extracted from them, and it has been used widely as one of the medications in the Ayurveda as well in India. But now recently also in the literature also, if we look into that, the berganine compound is extracted and has been used widely in many studies, specifically in the diabetes, because the berganine has a promising effects as anti-inflammatory. The most potent effect is anti-inflammatory. So most of the studies, they are using berganine to reduce the oxidative stress that is enhanced in diabetes. And actually, there is so many studies. Yeah, they show that it reduces the hyperglycemia in the diabetes. Yes, Dr. Vinod.
Yeah, no, I'm just interested looking at its anti-inflammatory properties, right? Whether
Yes.
it can be
Yeah.
used in infectious diseases. That's number
Correct.
one. That's why I asked. Yeah.
Yes.
Now you have done quite a bit of preclinical sort of a model, right? I mean, you use rats. Now, if you were to proceed this to clinical phase, now what kind of experiments would you improvise? Now, how would you change from your current setup? What
Yeah,
would you
we
look
need
at?
to. Yeah.
Yeah.
Yes, you're right.
Yeah.
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 The first thing is that if we are targeting this berganine into to go into the clinical trial or clinical phases, the first thing that we need to look at this, that whether the people's are whether this helps to alleviate the symptoms of diabetes itself. And then we need to think that okay, it can prove beneficial to the diabetic patients in terms of reducing oxidative stress, so that it can prevent the organ damage, because in the diabetes mellitus, organs damage is the most important thing that we need to look into that. So, and most of the patients, like male patients specifically, in the females, of course, we don't know much about the reproductive effects, but usually in the male patients, of course, the testicular damage is accelerated because of the oxidative stress, and then sperm motility, sperm viability, that is affected. So we can either we can, use it as an adjunct with the medications that the patients are using for the diabetes, because currently there are so many newer drugs also for the diabetes patients. So if we can use for that in as an adjunct to enhance the functions of the testes in the males, or the other functions like parameters, or the patients are willing to take for to enhance the sperm motility. So in the diabetes, then we can use, but of course we cannot use in normal individuals. But I have seen even berganin is used as a supplement as well. There are so many, if you Google it, we can find it that berganin is available as as a supplement, and people are taking it. So yeah.
Okay, nice. Thank you. Thank you.
Okay.
All right. Okay, thank you, Ms.
Thank you.
Huma, for your in-depth explanation. So now moving on to Mr. Mohammad Daniel Shafiq from UITM. Okay, please welcome.
Assalamualaikum and good day. My name is Daniel Shafi. Glaucoma is an optic neuropathy due to progressive degeneration of the retinal ganglion cell, which leads to irreversible blindness. The treatment target is to reduce the intraocular pressure, or IOP, which is the major risk factor of glaucoma. Increase in extracellular matrix, or ECM, deposition in the trabecular measure contributes to the aqueous humor outflow resistance, causing elevation of IOP and progression to glaucoma. Fibronectin, or FN, is the most abundant ECM in the human trabecular measure and found to be elevated in the trabecular measure of patients with open-angle glaucoma. Transresveratrol, or TR, is a natural compound with the ability to reduce the IOP in steroid-induced animal glaucoma model. We hypothesize that the IOP lowering effects of TR involves reduction of FN expression in steroid-treated human trabecular measured cells, or HTMC. Hence, this study aims to see the effect of TR on the FN expression in steroid-treated HTMC. HTMC were cultured to Passage 5 and divided into 5 groups. The first group, HTMC incubated with media only. The second group, HTMC with 0.1% DMSO. The third group, HTMC with 100 nanomolar dexamethasone. The fourth group, HTMC with 12.5 micromolar TR. And the fifth group, HTMC with both dexamethasone and TR. The cells were incubated for 7 days treatment. MTS assay was carried out first to assess the cell viability after the 7 days of treatment. And ELISA was performed to measure the protein expression of FN in the media of each group. The result of this study, there were no significant differences in the cell viability between the 5 groups. Besides that, FN expression was significantly higher in the media of HCMC treated with dexamethasone compared to cells incubated in media only. Whereas the TR-treated HCMC group has significantly lower FN level in its media than the dexamethasone-treated group. Adding 12.5 micromolar TR to dexamethasone-treated group for 7 days significantly reduced the FN expression. To recapitulate, a 7-day HCMC incubation with TR 12.5 micromolar with or without dexamethasone is safe and non-toxic. Furthermore, TR reduces FN expression in steroid-treated HCMC. That's all from me. Thank you.
Okay, Mohammad Daniel Shafiq, are you here?
Yes, I'm here.
Okay, thank you for the nice presentation. I like the way you talk. I like your poster. Okay, just one question from me. So, has there been no study done on resveratrol on glaucoma treatment before?
Yes,
has there been a study?
Yes, it has been done on animal study on the IOP on animal on the rats. So it shown to reduce the intraocular pressure, or IOP, in the rats. So my study is on the in vitro study.
All right, so in vitro study that has never been done before?
Yes.
Right. Okay. Interesting. So, because there have been many studies, right, that have done on resveratrol. So, how do you think that resveratrol works? What is the mechanism of action on this fibronectin that you look into? Can you explain further the mechanism?
So there was a previous study that showed TR reduced the expression of MMP, matrix melanoproteinases, in animal and also in the trabecular measure. So this MMP is the role is to degrade the ECM, such as this fibronectin. So the elevation of MMP by the transviral trough for the PSP study can be associated with my reduction of fibronectin expression in the tubercular measure.
All right. Okay. Thank you, Daniel. Okay. All right. Very well done. Okay. So next, we would like to invite Dr. Cornelia Wilson. Sorry, are you around here? Cornelia?
Yes, I'm here.
Okay, great. Okay, thank you. So shall we start with your presentation? Okay.
Hello, my name is Dr. Cornelia Wilson from Christchurch University. Here I present our work on black-spitting cobra, their extracellular vesicles, the biological function, and their interactions. The therapeutic properties have been characterized for a variety of animal venom components, including enzymes such as metalloproteases, thrombin-like serine proteases, and phospholipases, which are largely proteins with a role in venomation effects. More recently, extracellular vesicles, which are small vesicles released by cells, have been characterized in snake venoms as well as in venoms of other species. The black-necked spitting cobra, known as Niger venom, is unusual to other lapids as it consists mainly of cytotoxins. In this study, we've investigated whether EVs were present in the venom of Niger which was analysed with the aim of classification in a species which these EVs has not been extensively described. So in figure 1, we've analysed the size and concentration of snake EVs from Niger by nanoparticle tracking analysis and also electron microscopy. We demonstrate that Niger venom contains vesicle-like structures characteristic of EVs. We show that the snake venom EVs in figure 2 contributes to hemolysis effects in envenomation compared to the whole venom. Next, we investigate whether Niger venom EVs contained phospholipase activity, and we demonstrate that they significantly have a higher phospholipase activity compared to whole venom. We observed venom EVs to be taken up into mammalian cells. These EVs were found to inhibit lung cancer cell growth and cause apoptosis as seen in figure 4, and this actually contributes to the understanding of Niger venom envenomation and the profile of EV action in cell signaling and metabolic influence. Future work will be assessing the venom EV content and their mechanism of action on recipient cells.
Hi, Cornelia. Thank you.
Hello.
Thank you so much. It took some time for me to actually understand. It looks complex, but
it is,
wonderful study, wonderful study, especially considering that, EVs are, lately being mentioned a lot in cancer studies, right? And
exactly.
any particular reason for choosing this type of snake, whether, I don't know. And do you have any knowledge of EVs between snake venoms, whether, there are huge changes in terms of content, the molecules inside it, and can you elaborate a bit, please?
Yes, of course, yes. Thank you. Good afternoon, everyone. So we chose the Niger venom because we could actually harvest quite a lot when we were milking the snakes. The venom comes from a company that we work with, it's called VenomTech. And you can harvest around about 3 to 5 mils of venom. In fact, the venom is actually packed with extracellular vesicles. And we've actually studied venom from other species, so from scorpions and spiders. In particular, we've looked at venom from Loxosceles, which is a brown recluse spider. What's interesting about the extracellular vesicles is there's actually not a lot of studies done on extracellular vesicles from snakes or spiders. There's starting to be kind of more and more reports coming out. What's interesting is that there's kind of the content of those extracellular vesicles. They're essentially carrying many of the components that we've actually identified in the kind of whole venom. They're actually being kind of concentrated in the venom. So from our study, what we've seen is that when we kind of concentrate that venom, so when we can extract the extracellular vesicles using kind of typical techniques such as differential centrifugation,
what we see is those extracellular vesicles are actually much more potent than the full venom itself. So which kind of begs the question really why are extracellular vesicles present in the venom? And I think the role of these extracellular vesicles is maybe there's an evolution playing a part in this, because we see extracellular vesicles in all sorts of organisms. So we can see it in bacteria, in yeast, and they're there as a kind of communication network. Not only these vesicles, they can have, they can modulate the kind of genome that the cells that they kind of essentially enter. They are playing a mechanism, maybe allowing the particular venom to kind of take hold. We know that the extracellular vesicles are delivering a cargo, and that cargo is obviously having an effect. And obviously we were quite interested by lung cancer. So we chose obviously the lung cancer cells that we were looking at in the study. I hope that answers your question.
Have you, yeah, absolutely. Brilliant. Have you looked at the content of the cargo, maybe via LCMS or, the full content of the cargo, just to get an idea of the mechanism of it inducing, apoptosis in cancer cells or.
Yeah, so that's actually our next study. So we're just about to start to look at the content of the extracellular vesicles. There is one study which was published, I think last year or the year before, where they looked at other types of snake EVs, and they did see actually there was different components. So there was like neurotoxins in there. We have obviously phospholipase. Even components that kind of target particular receptors on cancer cells, so things like NGF and VEGF. So there's particular cargo. What would be nice to, if you work in the extracellular vesicle field, what people want to see when you characterize them is, okay, we can characterize them by the transmission EM, so we can say, okay, the size. We can also characterize them by using so essentially population, the size again, using the nanoparticle tracking. What would be really nice is maybe to see any kind of particular, we call exosomal proteins. So these are proteins which would be found in extracellular vesicles. And now that hasn't actually been done so far. So I think that would be interesting to look at, to see whether or not they have particular markers of extracellular vesicles.
That's just brilliant, Cornelia. Thank you so much. Thank you.
Thank you. Thank you.
Thank you, Dr. Cornelia, for your nice presentation, your poster. Very nice. Okay, so next presenter is Leong Sien from UPM Bintulu campus. Okay, let's hear his presentation. Sorry, her.
Okay, so I'm Associate Professor Dr. Leong Sien. So I'm from UPM Bintulu campus. So the title of my poster is the enhancement of the PM fruits extracts on the male fertility tested in rats. So my study aims to determine the effects of PM on the male fertility in adult rats. So methodology begins with the aqueous extractions of my PM fruits. Then experiment design started as 60 dolly adult male rats, 7 weeks old, weighing 200 to 250 grams, were randomly divided into 10 groups with 6 rats in each group. So a 2-by-5 experiment design with 2 supplementary periods was conducted. So the rats were treated with 5 different doses of 0.2 ml of PM, so as stated in the poster, for 3 and 7 weeks. So parameter measures are testis size, the testis volume, body weight, and also the testosterone level. So middle of my poster shows all the results. So based on the figure, you can see, so they are analyzed using the SPSS, shows the mean testis size was similar among the treatment groups. And then the forced feeding of the fruit extract protein, no significant change on the testosterone level, ranging from the 1.03 to 1.42 ng per ml, while the testis volume remained 2 ml throughout the study. Body weight was significantly highest in the rat treated with 240 mg per kg. Okay, so the rat treated with various dosage of PM did not produce any significant change in the plasma testosterone concentration and testis volume. Although the concentration and testosterone do not change significantly, but we believe it shows actually a percentile readings with those as shows in the figure. So this might be due to the dosage was not sufficient enough, and also the duration of supplement was not long enough to produce any significant effect. In conclusions, the effect was dose and time dependence, and the result shows that PM supplement significantly increased the fertility of rats, and the effect was dose and time dependence. So thank you so much. Okay, so I stop recording.
Okay, thank you very much, Dr. Leong. Are you around? Sorry, Dr. Leong Sien, are you around? Dr. Leong? Okay, I can't seem to find Dr. Leong here. Shall we move to the next presenter? Meena Selvanayagam from UPM. Okay, please welcome.
Good day everyone, my name is Meena Selvanayagam from Universiti Putra Malaysia, and my research is on the antioxidant properties of hot water extracts from Crescentia cujete leaf, bark and fruit. So calabash tree, also known as Crescentia cujete, previous studies were made on these plants on the terms of antioxidant activities as well as its bioactive compounds. However, limited evidence for the use of each part of this plant in terms of its antioxidant ability was found. For the results and discussion part, high amount of TPC was found in 50% ethanol bark extract and no phenolics were detected in fruit extracts as negative results were found. High TFC content was found in hot water leaf extracts and the lowest was found in 50% ethanol extracts. As for DPPH scavenging activity, highest percentage inhibition was shown in hot water leaf extracts having the lowest IC50 value. This indicates that high DPPH scavenging activity in hot water leaf extracts as well as high TFC content shows that polar phenolic compounds in the 50% ethanol bark extract may not contribute directly in the antioxidant activity. As for the ferric reducing power assay, high reduction ability was found in 50% ethanol fruit extract, which indicates that other compounds apart from antioxidants may be responsible for the oxidant reduction in the fruit extracts. To conclude, hot water extractions of Crescentia cujete leaf exhibits the highest antioxidant activity. And that's all from me. Thank you.
Hi, Meena. Thank you.
Yes. Thank you, Doctor.
Thank you for your good presentation. I don't have that many questions. Number
Okay.
one,
Sure.
what is the purpose apart from determining, the type of extraction and which parts which yields the highest activity? What is the purpose of
Yes.
this experiment? What is the purpose of this project? How do you bring forward next? Okay, you have found which part is giving the highest, extract. What are you going to do next?
Okay, so the main reason or the purpose of this project was comparing the hot water extract with the ethanol extraction, because in folklore, usually this sample they only use it in very indigenous, very countryside. They don't use it they only use it in very local environments. So and in that kind of environments, they boil the extract of the plant, whether it will be fruit or usually in fruit and leaves, and they will consume it. So in folklore, it is said it has a very high antioxidant activity. So using that as a purpose, I'm doing this experiment, this research, and there's very few studies on the antioxidant properties of each part of the plant. So I want to know, doing this study to know which part of the plant has high antioxidant properties, and do more studies on it. Yeah.
So what does this plant do exactly? Have you seen, are they using it because high antioxidant? Are they using it on cancer cells or cancer, animal models? Or, can you elaborate a bit on this plant and also its, use? Since it
Crescentia
has, high
cujete.
antioxidant.
Oh, yes, sure. Sure.
Yeah,
Okay,
sure. Yeah, please
Crescentia
continue. Yeah.
cujete. Crescentia cujete is actually locally in Malaysia. It is known as pokok labu. Pokok labu, also known as pumpkin, in a very simple way, and it's very rare, and usually it is found in mostly in Southern America or India, and it is said to have very high antioxidant, which is usually local people they consume it for cancer treatments. Yeah.
Okay, thank you. Thank you.
Okay. Oh, sure. Thank you.
Okay, I'm just gonna try once again. Dr. Leong Sien, are you around? I don't see you also in the attendees. Okay, all right. With that, we have come to the end of the session. Okay, thank you very much for still staying with us. Okay, in spite of the technical issue. And could we have quickly could all the presenters turn on the camera for a photo shoot? Just the presenters, please. Thank
And also
you.
judges.
Thank you for still being with us. Okay, yes. Okay. All right. Just take whoever are still around. Okay. 1, 2, 3. Smile. Another. Dot. 1, 2, 3, and smile. Okay. All right. Thank you very much again, everyone. It was a good day. Great presentation, everyone. And please take care. And don't forget to join us tomorrow. All right. Okay. Bye. Assalamualaikum. And have a
Thank you
good
so
evening.
much. Okay. Bye-bye.
Thank you. Take care, guys. Take care.
Thank
Okay.
you.
Thanks. Thanks, Dr. Binod.
Have a good
Thank you.
evening.
Thank you, Dr. Satirah. Yeah.